Okay. Okay, it looks like we've stabilized a number of people. Welcome, everybody. Good morning. It's my distinct honor and pleasure to introduce Mark Phillips, a candidate for a tenure-track assistant professor position in integrative biology. Mark actually began his academic career as a bioengineering student at the University of Florida, but upon taking an inspiring evolutionary biology course as a sophomore, switched into biology. He then went on to do a master's and PhD at UC Irvine in the lab of Michael Rose, where he obtained an NSF DDIG, and he finished his PhD in 2018. That year, he joined Molly Burke's lab as a postdoc here at OSU. He is now still in the lab as a postdoc, funded on an NSF postdoctoral research fellowship he was awarded. Yeah, we don't actually have the automatic mute on, so if people could mute when they come in, that would be good. Okay, where were we here? Mark is a computational evolutionary biologist and population genomicist. His research focuses on genetics of adaptation, and he uses experimental evolution, genomic sequencing, and bioinformatic tools in his research. So far, he has worked on model systems, but his future research will target genomics of evolution in natural populations. And even at this early stage, he has 15 publications. So with no further ado, Mark, take it from here.
Okay, thank you for that introduction, and thank you all for giving me the opportunity to present my work today on the genetics of adaptation in sexually reproducing populations. So before I get into my research, I just wanted to talk a little bit about the sorts of questions I'm interested in and how I approach those questions. So a lot of my work centers on using experimental evolution to answer questions about the process of adaptation. So for those of you unfamiliar, experimental evolution is essentially when you bring populations into the lab, subject them to some selection pressure of your choosing, and study how they change over time. And while this is a conceptually very simple approach, it's been used to great effect for decades to answer a number of fundamental questions in evolutionary biology. And here's a schematic showing one such experiment done by Diane Dodd in the 80s, where she brought fruit flies into the lab, split them between two treatments defined by different diets, allowed them to evolve for many generations. And then when she brought them back together, she was able to show there were signs of incipient speciation. So through this relatively simple experiment, she was able to derive insights into how ecological adaptation can drive the process of speciation. In more recent years, people have begun combining experimental evolution with next generation sequencing technologies. And this has allowed us to not only study how populations are changing phenotypically, but also looking at the underlying genetics driving those changes. And most of my work falls underneath this umbrella. So as people started doing these studies, one contrast became immediately clear. In asexual populations, adaptation was being primarily driven by new mutations that would increase in frequency until they fixed in the population. And when I say fix, I mean going to a frequency of 1. However, in sexual experiments, so a lot of these were done with Drosophila, adaptation was being more fueled by variants already present in the population. And these genetic variants would increase in frequency, but they wouldn't go all the way to fixation, which suggests a fundamental difference in the underlying dynamics. However, some people argue that perhaps this difference was more due to limitations of sexual experimental evolution studies. So unlike E. coli studies that characterize most of the asexual work that often have hundreds to thousands of generations of selection, massive population sizes, and often over 10 replicates, Studies with sexual species due to logistic constraints are usually much shorter in duration, have smaller population sizes, and fewer replicates. So you can make the case that perhaps mutation isn't a big factor in these studies because of the small population sizes, and we don't see fixation of beneficial alleles because they simply aren't long enough. So for my PhD, I focused primarily on this issue of experiments being too short. And this is work I did at UC Irvine, where I was primarily advised by Michael Rose, and Dr. Larry Muller served as a secondary advisor. So in this lab, there's an experimental radiation of Drosophila populations that dates back to the late 70s. And because this is such an old system, it actually does have populations that have been subjected to the same selection regime for hundreds of generations, as well as newly created populations that have dozens of generations under selection. And this is sort of a schematic of the system I'll sort of explain through the first half of my talk as I get to it. But yeah, in general, I took advantage of this historic system to address these questions about long-term versus short-term responses to selection. In terms of the system itself, it was created to study the evolutionary theory of aging. So this is based on work by William Hamilton done in the 60s, where he proposed that the ultimate reason for the physiological decline we associate with aging is because the force of natural selection acting on an individual varies over time. Specifically, it decreases over time. So one way to think of this is when you're in this area here, prior to the start of reproduction, natural selection is at its strongest, right? Because imagine if you had a lethal mutation that would kill an individual in this area, there's zero probability of that mutation making it to the next generation because the individual dies before they can reproduce. However, if you have a lethal mutation that's late acting, let's say it acts over here, natural selection won't be able to efficiently act against that mutation because by the time its impacts are felt, the individual has already reproduced. So the idea is that the reason things decline physiologically as they age is because of an accumulation of these deleterious mutations with late acting effects. And sort of the textbook example of this is Huntington's disease. So this is a dominant lethal mutation. And generally speaking, you would expect selection to be very efficient at acting against such an allele. However, because the age of onset is so late, the allele is usually passed on by the time people are hurt by its effects. So it persists in our populations at higher than expected rates. So to test this theory, one way to go about it was to see if you can select for populations that have increased longevity based on these ideas. And one way to do that is to basically delay the start of reproduction, which is represented by this B parameter. So if you're shifting this to the right and delaying the start of reproduction, you're basically increasing the amount of time where selection is acting at full power. So by having selection be efficient for a longer portion of the organism's lifetime at the population level over time, this should result in increased longevity. And it turns out this is actually quite easy to do in fruit fly populations. So in fruit fly labs, populations are often maintained in non-overlapping generation cycles, which means there's a specific reproductive window, so a specific time frame where population or eggs are collected to found the next generation. For instance, in the baseline populations for the Rose Lab system, only eggs laid on day 14 are used to found the next generation. So as far as natural selection is concerned, day 14 is the first age of reproduction or the start of reproduction. So if you were to shift this reproductive window to the right, you are essentially delaying the first stage of reproduction. And that's exactly what was done. And this was shifted all the way out to day 70. And then they looked at how did this impact patterns of longevity. And here we can see some results from that experiment where we have survivorship on the y-axis and time on the x-axis. So this is the survivorship curve for the baseline populations, and then this is the survivorship curve for the populations that were subjected to selection for delayed reproduction. And as you can see, the curve is shifted to the right, meaning the individuals in this population are living for a longer period of time on average than the baseline. So it's in accordance with the theoretical predictions. So from there, they decided to see, okay, if we can select for increased longevity based on these ideas, can we select for reduced longevity? And here it would be as simple as moving the start of reproduction to the left this time, right? Because if you were to move the start of reproduction to the left, you're basically decreasing the amount of time where natural selection acting on individuals is at maximum force. And as you might imagine, this was done once again by shifting the reproductive windows to the left. And they shifted it all the way back to day 9. And then again, looking at the survivorship curves, we see that in green, the survivorship curve for these populations where selection has been imposed for early reproduction, the curve is shifted to the left, meaning these individuals are living for a shorter period of time on average. So this altogether serves as a really powerful test of the evolutionary theory of aging. However, aspect of this system that I was able to use to my advantage is that these selection regimes create a gradient in generation times, in the sense that these populations on a 9-day cycle experience 3 generations per month. These populations on the baseline or normal 14-day cycle experience 2 generations per month, whereas these populations where their selection for delayed reproduction actually need over 2 months for a single generation. So we sort of have this natural gradient in generation times. So in terms of my, the first major study I'm going to talk about, I was focused primarily on characterizing the long-term responses to selection. So this is work that was published in Scientific Report in 2016, and I had 2 major questions. Does consistent directional selection drastically reduce variation in populations? And does the response to selection involve selective sweeps? And these 2 ideas are intimately related and hinge on this idea of selective sweeps. So to make sure everyone's on the same page, I'm going to explain what a selective sweep is. So we can imagine that these horizontal lines represent individuals in a population. So each one of us is a different individual. And then we're looking at a region of their genome. And then the circle represents alleles possessed by those individuals. We'll also imagine that the black circles are neutral alleles and the green circle is a beneficial allele. So based on the theory of natural selection, we would expect the frequency of the beneficial allele to increase over time until it goes to a frequency of 1, right, and fixes. However, it's important to consider that this, lost my cursor, this allele doesn't exist in isolation, right? It's on a specific genetic background and it's going to be linked to nearby variants in that background. So it's not simply the case that this allele will increase in frequency, the linked allele will also increase in frequency. So after the selective sweep event, so many generations later, you should see a pattern like this, where the beneficial allele and the neutrally linked allele have basically gone all the way to fixation and swept out all the other variants in the region. And this actually has a really distinct pattern if you're looking at genetic variation and how it varies across the genome. So imagine if you were looking at the chromosome where this region is located, you should see a pattern like this if you're looking at genetic diversity, where there's some sort of baseline level of genetic variation, but then when you get to the region where the positively selected site is located, you would see a depression in that invariation that goes all the way to 0. And you can imagine having these events across the genomes are going to have impacts on global levels of genetic variation if you have enough of them. However, as I mentioned at the start of the talk, people weren't really finding that particular pattern in a lot of the early sexual experimental evolution studies. Instead, they were seeing a pattern more like this, where you would get a depression associated with positively selected sites. However, these depressions wouldn't go all the way to 0. So as I mentioned, one explanation was that these experiments, which were often only a few dozen generations, weren't long enough. So here I want to see if after a much longer period of time, do we get complete fixation? And to jump right into the results, these are plots showing genetic diversity as measured by heterozygosity across the major chromosome arms for Drosophila. So each panel is a different chromosome arm, and then we have positions across the chromosome arms. And then 5 replicates are plotted on each panel. And these are replicates of the baseline populations, which at the time of sequencing had about 900 generations of selection. And as you can see, we do find these depressions in heterozygosity across all the replicates, across all the chromosome arms. So that's an indication that selection is acting on these populations. However, they're not going all the way to fixation. So they're not going all the way to zero. And I don't really have time to go into it, but I did extensive simulation work showing that 900 generations is in fact enough time for you to have fixation of beneficial alleles, given a lot of conventional models that most people sort of use to talk about these things. And in general, far more genetic variation is being maintained in these populations than we would expect based on conventional models. So that tells us something else is going on in the system. So in terms of the original questions I have, the answer to both is no. We don't have these selective sweeps, and in general, far more variation is being maintained than conventional models would suggest. So for my the next study I'm going to talk about, I wanted to do more in terms of comparing short versus long-term responses to selection and seeing which allowed me to think about some of the implications of my previous findings. So this is work that was done in collaboration with Joe Graves at North Carolina A&T, and he and I are co-authors on the manuscript based on this work that was published in Molecular Biology and Evolution. So here I have two big questions. The first is how repeatable is adaptation? And the second is how fast is adaptation? And both of these questions hinge on this hypothesis that I have that adaptation should be really fast and highly repeatable if it's different, because we know it's being driven by standing genetic variation. And based on my previous results, we know standing genetic variation is being maintained in populations. So the idea is it's rapid because there's no need to wait for new mutations, and it should be repeatable because the same core materials driving adaptation should always be present. So regardless of when you derive your populations or how many replicates you derive from a single ancestor, so all of this hinges on the idea of coming from a single ancestral source. Things should proceed in a relatively deterministic way because you're working with the same materials. So to look at this idea, I focused on three of our treatments. So the A and B, so these are two treatments I talked about before. So the A's would be our 9-day cycle, the B's would be our 14-day cycle. And then I use the C's, which are on a 28-day cycle. And you can think of these as a milder version of the 70-day cycle populations I talked about. However, they still have increased longevity relative to the A and B populations. So these are sort of the three we're focusing on. So looking at a schematic for the actual experiment, we basically have two versions of each type of population. So we have the long-standing or the old versions, so old A, B, and C, where generations under selection range from around 300 on the low end with the old Cs to around 840 on the high end with the Bs. And then we made brand new versions of these populations, all derived from the same ancestral source. And these new versions have from between 37 generations of selection in the case of the new Cs, all the way to 146 in the new As. So looking across the system, we have a pretty nice gradient in generations under selection, so we can sort of answer these questions about short versus long-term responses. And then the entire system is replicated 5 times. So 30 populations split between 3 treatments, and then new and old for each treatment. So I don't have time to go through the phenotypic results. I'll just say we've characterized these populations extensively with regards to a number of life history phenotypes and stress-resistant phenotypes. And in all of these cases, we find phenotypic convergence between newly derived and long-standing populations subjected to the same selection regimes and differentiation between selection regimes. So this is essentially telling us that within dozens of generations, you're getting the same magnitude of phenotypic adaptation as you're getting in hundreds of generations, which means that a phenotypic adaptation is incredibly fast, right? Because it's happening on that time scale. So for me, the question was, is this also matched by genomic convergence? Because at this point, we know adaptation is really fast. So, but is it proceeding in the same manner in each case? So to address that question, I look at patterns of SNP differentiation between the different groups of populations. So here's some plots showing that, and I'll just take a little bit of time to explain these plots because I'm going to be showing similar plots throughout the rest of the talk. On the y-axis, we have log transformed p-values. On the x-axis, we're going across the genome, so we have the chromosome arms. And then these black dots represent the results of statistical tests comparing SNP frequencies between groups of populations. So for instance, in this panel, we're comparing the new A populations to the old A populations. And then the red line is a permutation-derived significance threshold. So any points above the line would be SNPs that are significantly differentiated between the two groups. And as you can see, there's no SNP differentiation between the new A's and the old A's. There's also no SNP differentiation between the new B's and the old B's, or the new C's and old C's. So this tells us that these populations not only have they converged phenotypically, they've also converged genomically. So in total, it shows that adaptation is rapid and it's happening in the same manner at the genetic level, so it's repeatable. And just to make this a little bit more convincing, I'll show the between treatment comparisons. So it's not simply the case that the system as a whole is all the same genetically and say the changes we're observing are due to plasticity, because we can see that if we compare A to B, A to C, or B to C, we do find regions that are differentiated between treatments. Though notably, there's less differentiation between B and C, and we think this has to do with the intensity of selection. For instance, we know the A selection is very intense, while the B and C selection are less intense. So in terms of my conclusions, I find that in accordance with my hypothesis, adaptation is highly repeatable and it's really fast, so sort of supported. And this is the last bit I'll talk about in terms of my dissertation work. And just in general, looking across my dissertation, I do a lot of stuff like this where I show that conventional models aren't working and I show implications for them not working. However, I don't necessarily do a lot in terms of explaining what's actually going on in these systems, like what are the mechanisms driving these patterns. So for my postdoc, that's what I wanted to do. And to do that, I actually made the switch from working with fruit flies to yeast. So yeast are a really nice system to answer these dynamic questions because they're eukaryotic and capable of sexual recombination, but they're also a microbial system. So you can get a lot of generations in a very short period of time. And you can have really big population sizes. So you're also able to address that issue of, okay, now that we have these big population sizes, mutation might be more of a relevant force. So next, I'm going to talk about a project I did in this system looking at the value of high resolution allele frequency trajectories. So this is work that is based on a study by my postdoctoral advisor, Molly Burke, that was published in 2014. So in her study, she had 4 very different haploid strains. She combined them to make a big recombinant population. So this population now has a lot of diversity because it was created using very dissimilar strains. And then from this source population, she derived 12 replicates. And then these replicates were subjected to a selection regime defined by forced outcrossing for 18 weeks. And here, 18 weeks corresponds to about 540 generations. And the populations were sequenced at 4 time points as indicated by the lines for DNA sequencing. So the idea is to find regions of the genome that are increasing consistently across the replicates over this time frame, because that would be suggestive of selection acting on those regions, right? And the major finding from her study was adaptation was still primarily being driven by standing genetic variation, even though you had a real potential for mutation to be a factor here. So we can sort of look at the big results. So here again, we have negative log p-values going across the chromosome, and these are results of her test looking for consistent increases in SNP frequencies, and a number of candidate regions appear. And we know this is from standing genetic variation because these regions that are increasing are all present in generation 0. And there's no evidence for, say, an allele appearing in the middle of the experiment and then increasing like you would expect if mutation was a driving force. So again, even when you have a lot of time and a lot of individuals, standing genetic variation is providing most of the material for adaptation in sexual populations. So I kind of went back to this study in some work that was recently published in Molecular Ecology, where I asked, okay, can we learn anything new from having more time points? So we went back to the freezers and basically sequenced a bunch of other time points. So instead of just the 4 she had, now we have all of these other time points as indicated by the dashed lines. And here my big question was, does increasing the number of time points sampled offer deeper insights into evolutionary dynamics? So the way I went about this was, I would say do an analysis looking for candidate sites using all of the data, so all of the time points, and then I would use self-organizing map to characterize the SNP frequency trajectories present among those candidate sites. And then I would do the same thing downsampling to fewer time points. So in terms of what the downsampling looks like, first I did the analysis, like I said, with all of the time points. Then I did a version with 13 time points, again, showed by the dashed lines, a version with 8 time points. And here it's important to notice as I downsample, I'm always sampling across the entire evolutionary trajectory. It's not the case that I'm, say, just using the first 8 time points. I'm sampling across the experiment. And then back to the original 4 time points. So here are the results from that analysis. So I'll just explain these figures. So we'll start with the top panel. So this is showing the, the trajectory patterns present among the candidate SNPs from the analysis using 4 time points, and the patterns are only using 4 time points. So for instance, among those candidates SNPs, we have 1,659 SNPs that show this type of trajectory pattern. We have 742 that show this pattern, and we have 1,484 that show this pattern. And then similarly, if we have 8 candidate sites, if I use 8 time points, I have these patterns. If I use 13 time points, I have these patterns. And lastly, 17, so this is the total, all of the time points, I have these patterns. So as you would expect, as you increase the number of time points, you get higher resolution trajectories. It's also worth noting that some patterns that are present when we have 8 or more time points, notably this pattern where there's an increase and then a plateau, is not present when we only have 4 time points. So you do gain some new information into the underlying dynamics. You don't with only 4 time points. So next we can talk a little bit about what these patterns mean. So these are the sort of highest resolution patterns from when we use all of the time points. So in category 1, we're seeing a pattern characterized by little initial change followed by an increase. In category 2, we're seeing a continuous increase. And in category 3, we're seeing an initial increase followed by a plateau. And here it's important to note that this plateau is not due to fixation. So these, basically these SNPs could keep increasing in frequency, but they don't. So in terms of what these patterns might mean, I sort of speculate on what the underlying genetic models that would produce these would look like. For instance, this category 2 pattern is consistent with a conventional model where you have some constant selective benefit or some constant selection coefficient, and then the allele simply increases in proportion to that benefit. However, I would argue that these category 1 and category 3 patterns are perhaps more context-dependent patterns. For instance, I think this category 1 pattern is suggestive of positive epistatic interactions. And just as a reminder, epistasis is when we have gene-by-gene interactions. So you can imagine you have an allele that by itself is effectively neutral. However, when paired with another allele, it has some benefit. So you could see a scenario where you have this allele neutral over here, but then say as a category 2 site that it has an interaction with increases, you start seeing those pairings more often. As a result of that pairing, this allele is now beneficial, so it increases in frequency. So an increase driven by a positive epistatic interaction. The category 3 pattern, I think, is perhaps suggestive of genetic redundancies. And this is the idea that if you have multiple alleles that contribute to the fitness or the phenotype in the same way, you don't need both of them to fix in the population for the population to reach an optimal adaptive state. Instead, you only need the redundant alleles to be common enough that every individual has one or the other, right? Because then you have coverage in the population. So if everyone has A or B, everyone is able to get to the optimal fitness if A or B contribute in the same way. So that's sort of where I'll leave off for this study in terms of what I want to talk about here. And then the answer to this question is yes. By having these higher resolution trajectories, we were able to offer some deeper insights into the mechanisms of what's going on in terms of the underlying genetic model. So next, I wanted to talk about some ongoing projects and some future directions. Let's get some water. So this is the work I received my NSF fellowship to complete. And I'll also mention the experimental phase of this work is now complete, and we're currently doing all of the sequencing. And the experimental phase was completed with the help of 3 students recruited through the LSAMP organization on campus. So in this experiment, I'm asking, how does the intensity of selection impact the genomic response to selection? So the idea is I want to look at moderate versus strong and see how things change. So I do a relatively simple experiment where I have a set of control populations, a set of populations subjected to moderate ethanol stress, and a set of populations subjected to high ethanol stress, and then sort of seeing what the differences are. And here I have 2 primary hypotheses that I'll outline. So the first hypothesis is that in both selection regimes, you're getting the same targets of selection. The same targets are being, or the same alleles are being targeted by selection, but you'll get bigger changes when selection is more intense. So this is sort of a diagram of what that might look like, where we have red, green, and blue alleles in both. And then we have allele frequency on the y-axis, time on the x. So we're seeing how they change over time. And we'll assume these are beneficial in both sets. So the idea is when selection is moderate, you get an upward shift in the beneficial alleles, but you're able to get to the new optimum phenotypic state without them all fixing, because selection is relatively mild, so you don't have to evolve that far to get there in a sense. However, when selection is intense, you need to evolve further to get to that optimal adaptive state. So then the frequencies will increase all the way until they fix in the population. And this could potentially have some power to explain why we so often don't see fixation in studies. And here the answer would be, our selection pressures aren't intense enough in a lot of these studies, right? So this is some hypothesis 1. And this has a number of implications for the underlying genetics, and in principle assumes a very simple genetic landscape where things are largely additive. A second hypothesis, and this is one I'm really interested in, is that when you have different intensities of selection, you could have different targets of selection due to widespread pleiotropy. So as a reminder, pleiotropy is when you have a gene or a variant or an allele affecting multiple phenotypes. So here we can imagine that in the intense selection regime, the purple and blue alleles are the beneficial alleles, and they increase over time until they fix. However, we could also imagine that these alleles have some trade-off, right? So even though you're gaining ethanol resistance through them, you might be hurting some other phenotype. They're only beneficial in this context because ethanol stress is such a pressing concern. However, if you were to have a scenario where ethanol stress is less intense, these alleles would no longer be favored because they have such heavy costs or trade-offs associated with them. And instead, you might get 2 other alleles. So here are the green and the red that are beneficial, that don't have costly trade-offs associated with them. And if we were to see this pattern again, it would be suggestive of widespread pleiotropy, which would tell us something about the underlying genetic models at play in these systems. So from there, I'll just talk a little bit about the direction I want my own lab to go in. So and for me, I'm interested in, okay, how do we go about extending findings from experimental evolution to real populations in useful ways? And I was actually invited to write a perspective on this, which was recently published in Molecular Ecology. And sort of two big goals of this would be, could we use these results in models derived from experimental evolution to assess the evolutionary potential of natural populations and to predict how real populations will respond to certain selective pressures? In this diagram here, it gives a context of how that's useful. So here we're seeing sort of or an outline of how we can go about addressing changes associated with climate change and coming up with management strategies. So obviously a big part of preparing for climate change and adjusting to it is studying how things are going to change in terms of abiotic conditions, right? Like temperature growing up and all of these other things ecologists usually talk about. But however, another important component is how are the actual populations going to change and evolve in response to that? Because you also need to take that into account as you come up with management strategies. So knowing something about the evolutionary potential of populations could be very useful in this context. However, I think there's some big challenges to this right now, looking at the current state of the field. So a big one is if you look across, and this, again, I'm mostly talking about studies with sexually reproducing organisms, is there are a lot of inconsistencies. I would say more or less all studies agree that adaptation is being primarily driven by standing genetic variation. However, when it comes to things like the role of evolutionary history, evolutionary repeatability, and so on, there are discrepancies. Some studies find history matters, some find history don't. And I think a lot of this has to do with differences in the way studies are conducted. So in terms of how they apply selection pressures, what pressures they apply, how long are experiments, population sizes, etc. So I think clearing up some of that noise is an essential step if we want to make robust models from these findings. Another more fundamental issue is that most studies are geared towards genotype to phenotype mapping. So this comes with a lot of specific recommendations. So some of them I think are just universally beneficial. For instance, one of the biggest recommendations is to have as many replicates as you can. That's always going to be helpful because it's always going to give you more statistical power. However, another recommendation is that you should maximize population sizes on levels of starting genetic variation, which theoretically has been shown to be useful for genotype to phenotype mapping, but I think it makes it difficult to extend those findings to real populations, which don't necessarily have maximized population sizes and genetic variation. For instance, I think it's unlikely those results from studies that maximize those parameters would be representative of, say, a population that's recently experienced a bottleneck. So to move towards addressing these issues, I plan to start with looking at, or sort of a retrospective look, going back to past studies and doing meta-analysis. So some of this, I think a really important step would be standardizing data processing and analysis, because here we have a list of 15 commonly used tests across experimental evolution studies. And I think the fact we have 15 commonly used tests is somewhat problematic in the sense that that could be compared or creating artifacts in interpretation of the genetics of adaptation. And then once we sort of control for those things, looking at what we can pull out about how duration, population size, selection pressures, and so on impact our conclusions about the genetics of adaptation. Next, I would want to do explicit, or I'd want my lab to do explicit experiments testing how different population genetic parameters and experimental parameters interact to impact the genomic response to selection. So this would be, the experiment I'm doing now is kind of a precursor to some of that. However, I'm sort of envisioning experiments where instead of just varying the intensity of selection, we could also have treatments with different levels of starting genetic variation, treatments with different population sizes. Even in terms of how we apply selection, we could have fluctuating selection, whether that's through some pattern or random and so on. So basically trying to cover a broader range of scenarios that could be representative of what we find in nature. And I would primarily be doing this work with fruit flies. However, I do have interest in working with other model organisms and perhaps non-model organisms through collaborations with other researchers. And lastly, as we do all of this work and get a better understanding of the underlying genetics in terms of how important these factors like redundancy, epistasis, pleiotropy, and even balancing selection are, we can start generating robust models that have hopefully some real predictive power of how populations will adapt and change over time. So with that, I'll end the research portion of my talk and talk a little bit about the diversity and inclusion work, my plans for that, and what I've done in the past. So just a little bit about myself. I was born in Guyana, and most of my childhood was spent between there and Trinidad. And then I moved to Florida in the early 2000s. And that came with a lot of challenges in terms of adapting to a new country and culture and so on. But I think one big challenge was obviously learning how to navigate the American education system to get where I wanted to be, which was been a scientist for more or less my whole life. And I think a big part of the reason I was able to get here was along the way, different people gave me advice and told me how to do these things, right? So like when I started undergraduate, I didn't know the importance of doing undergraduate research because I didn't necessarily have parents or anyone to tell me that's something you need to do if you want to go to grad school. So having professors and people help me has been a tremendous asset. So I've always felt a need to kind of pay that forward. So when I was a graduate student, this specifically during my second year of graduate student, I started a program with some of my lab mates where we would go to Santa Ana High School, which is a minority serving institution in Southern California, and we would work with their biology classes. And with these classes, we would do experiments to reinforce various concepts they learned in class. And we would also have sessions dedicated to talking to them about careers in science, answering questions about college applications, what you do in college, what you do in grad school, and so on. And we did this for over the course of 4 to 6 weeks every year for the entire time I was in graduate school. And at least as of 2009, other people have been continuing the program. So as I start my own lab, I would be interested in doing this sort of work where outreach is done to underserved and underrepresented communities to actually get them engaged in the scientific process, and also providing information to people who might not have immediate sources of that, like I did. And next, I do also plan to be intentional about recruiting students from underrepresented backgrounds for my lab. So in terms of my lab structure, I'm imagining primarily a computational lab with some experimental components. So most of the computational work would be done by postdocs and graduate students, as I'm able to recruit and fund either. And a lot of the experimental work, there would be opportunities for undergraduate researchers to help. So as I talked about in my ethanol project, I recruited my assistants through the Lewis-Stokes Alliance for Minority Participation. And while I've been a postdoc at OSU, I've also interacted with other groups on campus like SACNAS and EMUS. So I think these would be a good place to recruit students from underrepresented groups. And in general, just more broadly, I plan to establish connections to minority-serving universities as places to advertise and try to recruit graduate students for my own lab. Here, I already have some connections, in particular to the HBCU system, where one of my longtime research collaborators and mentors, Joseph Graves, is at North Carolina A&T. He himself has done a lot of work in promoting diversity in STEM. And actually, as a graduate student, I contributed to a program where students from HBCUs were hosted at R1 institutions to complete summer projects as a means of long-term recruitment. So those are the kind of programs and connections I'll try to make over time to basically recruit more diverse candidates for my lab. And with that, I'll end my talk on some acknowledgements. So these are various people I've worked with over the years and my current funding sources. Thank you.
Thank you, Mark. Give you a round of applause. Okay, we'll do questions now with raised hands. Oh, we've got some clapping too. I didn't know you could do that in Zoom. Let's see, Anne Devon Song.
Hi, Mark, can you hear me?
Yeah.
Yeah, that was an awesome talk. Thank you for waking us
Oh,
up.
actually, I can't hear you now.
How is it now?
It's very quiet, Anne.
Yeah.
I'm going to type it in.
Okay. I'll look in the chat. Okay. Can you see that, Mark, or should I read that to you? I'll read it for everybody. The Wald-Vogel diagram is really exciting. I wonder how you would explain to an ecologist how to go from a controlled lab environment and apply that to changing environments and ecosystems with so much stochastic variation and selection pressure. Do these systems allow for some combination of experimental lab data with field data, for example? Or what sort of leaps do you have to make when interpreting natural system with experimental results?
Yeah, so that's a really good question. So obviously part of it, I think the lab component, you're going to be, you essentially have to assume that there's some key parameters that you can capture of the natural population, that once you capture those parameters, your findings will be at least in part representative of natural populations. So that is a little bit of a leap of faith, but I do think there's a role for incorporating or data from real populations as a way of validating models you derive from or you build based on your work in laboratory populations. And I think that's sort of how you make those connections. Because essentially what I'm working with in the lab is just time series data. So there are data sets of people doing time series work in real populations. So I think those are good sources of validation. And hopefully you can find some way to translate though. I'll always acknowledge there's some possibility you can't translate it, at which point I'm a failure, so that's okay.
Okay. I have a question, Mark.
Mm-hmm.
So the category 1, category 2, category 3 that you showed, where you had hypotheses as to your explanations as to what each of those represented. If you were to look, do you actually already have the data in the sequencing results that you've got, in the genomic sequencing results that you have, that would allow you to tease some of this apart? Like, could you actually take all the category 1 loci and look at the background, the rest of the backgrounds that those are in? And does that allow, I mean, I haven't really thought about it in detail, but could you actually tease out what's going on by looking at the context?
Yeah, so I think that's one idea that would be potentially possible if you could make clear associations on that front. Though another idea I've sort of thought of more recently that we didn't do for this paper that I think would be potentially effective is breaking the experiment into chunks and then evaluating selection coefficients, because you can estimate those from time series data. So you can actually see if the selection coefficients are changing depending phase of experiments, which would be suggestive of that turning off and on kind of dynamic.
Right.
I do think long-term, I think experimentally, this is a really good one you could target, because you're able to control the starting levels of variation when you make these populations, because you're combining strains, right? So if you combine those strains in different proportions, you can actually control what's starting at high and low frequencies. So if you do the sort of analysis you described and combine that with an empirical test of it, I think that would be a really strong way to test this.
Okay, thank you. Mark Novak.
Yeah, I had a question about this exact slide as well. Sorry I missed it, but could you describe to me what the y-axis is? And then I have some follow-up questions, obviously.
Yeah, so the y-axis isn't that meaningful because when you feed the data into this algorithm, you have to normalize it. So that's just the scaling of that.
But I can interpret it as the like fixation. Basically, the saturation on the right is fixation?
Yeah, if it were to get all the way up, that would be around fixation. Yeah, it's just not on a 0 to 1 scale because of that normalization procedure.
Okay, and could you what I'm struggling with is how you got categories from continuous variation. Because right, these dynamics, must be continuous. Some, like category 1, took a long time to take off. Others took off pretty quickly, and everything in between. So there must be some cutoff for what puts a particular pattern or a particular trajectory into a fixed category.
Okay, yeah, so the method I use for this is self-organizing maps. So this essentially you're feeding in your different trajectories, so any sort of time series data, and it's pulling out the features it can't, so basically reducing the dimensionality to give you these representations. And it uses a competitive algorithm to figure out which are the best maps to represent what you have. In some ways, it's similar to a PCA. It just has some supposed benefits if you have noise in your data. And I do think there's a lot of potential for noise since this is sequence data where you're going to have sampling issues, sequencing error, and so on.
So it's not just stochasticity that you have to end up with some trajectories that end up with category 1, and you have to, just by the nature of the data, end up with some categories if you run them long enough that go to fixation.
Yeah, so I actually can, because I did run this on random collections of data to show that. So here we can see versions on the right, or on the left, we would have the versions on the candidates, and then on the versions, we have the random SNPs. So it's not the case you always get the,
Thank you.
Virginia.
Thanks, Mark. That was a great talk. This is more of a planning question. I was fascinated by the table that you listed of all the models for that you plan to sort of try to weed out or focus on driving the field forward in a more organized way. It seems like this is an opportunity for the field of evolutionary biology to cooperate in a mission-driven way towards helping with climate change prediction. And my question for you is that being talked about? And, you know, have you thought about how you, Mark Phillips, can make that contribution maybe as a young leader in trying to bring the field together to work together towards this mission?
Yeah, so I would say it's starting to be talked about in limited capacities, but primarily just in terms of people trying to bring a population in the lab and expose it to a higher temperature and seeing what happens. So I think my contribution to where this is going is more in terms of advocating for being really thoughtful about how you do that and how you're able to extrapolate those findings. And I do think there's an opportunity for leadership there, because I think most studies, and there are exceptions, but people tend to take their results on face value, right? You bring your thing into the lab, you make it warmer, then you say that's your finding, where I don't necessarily think that's always a useful strategy.
Great, thanks.
Okay. Any more questions? So I have another one, Mark. So I'm thinking about this in the context of the beetles that I work on, and a lot of them, there's a whole group that are associated with snowfields, high elevation snowfields. Mm-hmm. And we don't know anything about population sizes, but we do know that those snowfields are shrinking in size, and they're effectively, the organisms are, as these snowfields are of course high elevation. As those snowfields shrink, the beetles are believed to eventually be sort of pushed off the top of the mountain. And I'm thinking about what it would take to learn something about the genomics of adaptation to these cold adapted beetles in natural populations in which it would be very difficult to bring them into the lab. They have year-long life cycles. Are we like decades off doing anything with this?
I mean, go ahead.
Yeah.
Yeah, so I guess my way of addressing that would be in a very broad scale way, thinking about what's happening in those populations. Because from the description you're giving me, it's one, a selective pressure is being imposed on them, right? Or a change. Yeah. But also it sounds like their population sizes are being crammed down potentially as they're being forced.
Potentially.
So I think there might be some value in, say, experiments where you have population size change or being reduced over time as a treatment, plus selection and sort of different combinations to tease that apart, and seeing how that affects evolutionary potential. Because I think one possibility is because you're having selection plus this reduction in population size, that might reduce the probability of them being able to adapt because drug is going to overwhelm.
Right.
So I think that's the scale we can do in the near term. I do think in more, if you want to get to the point where you're predicting which alleles might respond to selection, I think that's where we're
longer. Yeah. Okay, so we've got a question from Adam in the chat. In the spirit of extrapolating from genotype to phenotype, as well as from the lab to natural populations, what's the role of epigenetic mechanisms in this future research program?
Yeah, so I do think that's something to consider. And I didn't mention this, but in the ethanol paper I'm doing right now, or the ethanol project, we are looking at changes in gene expression. So part of this will be figuring out how much, because I think in a total way of, you have your population, you impose selection, and then it has to move to some new optimum, right? So epigenetic factors, so plasticity is gonna get you part of the way there. And then the rest of it is going to have to come from genetic changes. So I do think thinking about both of those components and how they work together is important. And that's actually part of the intensity of selection idea. Because you can imagine if selection is less intense, maybe epigenetics gets you pretty close to the new optimum, right? Because you don't have to travel that far. However, the farther you go, maybe epigenetics become less of a factor.
Thank you. Francis.
Yeah, hey, Martin, a wonderful talk. Thank you so much. In the spirit of kind of looking at this, how we traverse that experimental to observational landscape or chasm, this is stating the obvious, but nature is full of natural, of selection events. We have heat waves, we have hypoxia, and we have chronic stress that go away in my world. What are the opportunities to take the work that you do and apply it into the field to say, aha, this is the signature of selection, strong or weak. This is the signature of adaptation, strong or weak. What's your flavor on that?
Yeah, so different way. I sort of have some different thoughts on why I think stuff like that's interesting. So one idea is, so a lot of the work on scanning for selection or detecting selection in natural populations hinges on basically looking at the allele frequency distributions, right? So that's your evidence of selection. If you have some skew in the distribution of allele frequencies in a region relative to the background. And I think that has potentially a lot of power to figure out early on what's responding to selection. So let's say you have your system that's being perturbed in some way. You can do those scans and try to figure out, okay, what's showing early signs of responding. However, I think the value of experimental work there is not a lot of people have done work trying to validate those approaches in experimental work, which I think could be really valuable, because in experimental work, you actually know what's happening, selection is being imposed. So I think that way you can validate those scanning methods a little better and then maybe use them a little bit more effectively in your context. And then I just think in general, having time series data from the sorts of situations you're describing could just be really powerful in terms of validating models and allowing us to contextualize laboratory findings.
Great, thank you.
I think that any last really quick question? Well, if not, I'd like to thank anybody who wants to unmute Ken, and let's give Mark a round of applause for a really great talk. Thank you very much, Mark.